Good morning friends. Today we are going to discuss about gene transfer. Here gene transfer we are going to discuss about definition, significations and types. Types we are going to talk about natural gene transfer and artificial gene transfer. Gene transfer is a technique to transfer gene efficiently and stable into this cell or in an organism. Or else simply when you say alter this organisms or changing this organism. With similar kinds of gene transfer methods, genetic engineering and recombinant technology. Why? To make economically important plant species or microorganism, to treat diseases, to monitor the progenies, therapy treatment of cancer like other diseases. So gene transfer it is naturally occur in Agrobacterium tumefaciens to make genetically modified grafts. What are else required for the gene transfer? Vector, plasmid, virus and physical and chemical techniques are used as a vehicle to transfer the gene. So it is a mode of gene transfer. Based on this they are divided into two categories. One is natural gene transfer, another one is artificial gene transfer. So based on this mode, natural gene transfer, which is used natural agents like bacteria, cells, okay, and DNA, other material which is used. But there we are going to see bacterial transformation, conjugation, page retransduction, and this retroviral transduction, transposition, Agrobacterium tumefaciens mediated gene transfer. When you come to this artificial method of gene transfer, here it is an exogenous gene transmitted into this another cell. It is artificial method either done by physical method or chemical method. Physical method we are going to see electrophoration, bioelastic or particle bombardment, gene gun, protoplasm fusion and micro injection. Chemical method we are going to liposome mediated gene transfer, calcium phosphate mediated gene transfer, TAE textron mediated transfer and gene transfer using polyethylene glycol. So it is based on this mode we have seen it. Now based on this which organism it's affect, they are divided into two categories. One is horizontal gene transfer, another one is vertical gene transfer. Horizontal gene transfer, it is occur between two unrelated individual or an organism is called as horizontal gene transfer or else lateral gene transfer method. It is very commonly occur in prokaryotic and eukaryotic. So where it originates from antibiotic resistance in bacteria is the best example for horizontal gene transfer. So when you come to this vertical gene transfer, so vertical gene transfer it transmits from parent to offspring or genetic material transfer from one gene to another gene or it's one cell to another cell or one organism to another organism is called as vertical gene transfer. What are the steps which is involved in the gene transfer? First, isolation of gene of interest. Second thing is selection of plasmid. Third one is restriction of DNA. Third one and fourth one is insertion of DNA into plasmid. And fifth one is selection of gene transfer techniques. And finally, say insertion of RNA and cell culture and injection of to this target organism. This is a Agrobacterium tumefaciens infected things. So this is root infected. So it causes crown gall disease. So this is Agrobacterium tumefaciens crown gall disease which affected one. This is a bacteria. So it is a soil bacteria gram negative one. So they have a TI plasmid, tumor inducing plasmid. They have a several regions, DNA regions and virulence regions and origin of replications. So there it is used for this transmission for transferring genes from one plasmid to another plasmid. So this is a common how the Agrobacterium transfer into this tDNA2 in plant cell which is shown here. So this is a method which is natural method which is involved gene transfer. So it is a TA plasmid transferred into this plant cell, then integrated into plant DNA, host DNA, then after integrated it multiply. Then after multiplication it converted into this plant grown in the culture medium, then it is plant bearing gene into new traits. This is a natural gene transfer method. When we come to this artificial gene transfer, either we can do through this bioelastic or electrophoration or particle bombardment. So we have to select the tree or explant, so which is converted into this method. We have to bioelastic or it is electroporation through we are transferring this Actobacter tumefaciens. So when we insert into this plant cell, then we are allowed to grow in the either organogenesis or embryogenesis, and further it's developed into the state. This is a gene transfer technique. Thank you.